Okay, so today I would like to show you our work on cell composition between normal and transformed epithelial cells in mammals. In my laboratory, we are interested in the initial step of calcinogenesis. In a steady status in epithelial tissues, each epithelial cell is connected via tight cell-cell contacts to form monolayered or multilayered epithelium. And in this community, they somehow communicate with each other via cell-cell contacts or soluble ligands. So they know how to behave properly in their own community. So they live together peacefully and happily. But one day, oncogenic transformation occurs in a single cells. Yeah? And yesterday, this guy was a normal and good neighbor, but today suddenly become a bit strange. Then question is, what can happen? For example, these surrounding normal cells, are they able to realize this change and somehow try to respond to this guy? Or in vice versa, this strange guy may start to intimidate the surrounding neighbors. Or they may just neglect each other. So in my laboratory, by using mammalian cell culture system and also zebrafish and mouse in vivo model system, we are examining the interaction between normal and various types of transformed cells. Then, because of the work of us and others, we found that cell composition occurs between normal and transformed epithelial cells in mammals. And the cell composition is a process by which two cells with different property compete with each other for survival. So let me show you one example, cell composition between normal and less transformed epithelial cells. So to examine the interaction between normal and less transformed epithelial cells, we use MDCK epithelial cells first. So this MDCK cell, this is a non-transformed type of epithelial cells from dog kidney. Then we stably express GFP tagged RAS-V12. So this is the oncogenic form of RAS in a tetracycline inducible manner. And because we use TET-on system, so when cells were incubated in the absence of tetracycline, GFP RAS-V12 expression did not happen, and cells shows typical epithelial morphology with tight cell-cell contact. But upon addition of tetracycline, after 2 or 3 hours, then GFP RAS-V12 expression starts to be seen, and after 24 hours, cells lose cell-cell contact and start to scatter like this. So in this culture system, by addition of tetracycline, we could induce such a transformation phenotype. Then, by using this cell culture system, we designed the experiment as follows. So first, RAS cells were cultured in the absence of tetracycline, so RAS expression is off. But to mark these cells, we use CMPTX dye. So this is a plasma membrane permeable red fluorescent dye, and stain them in red. Then we mix with normal MDCK cells at a ratio of 1 and 100. Then the mixture of cells were cultured on top of collagen gel until they form a monolayer of epithelial cell sheet. Then we add tetracycline. So under this condition, very small number of RAS cells, like 1 cell or 2 cells, start to express RAS-V12 while being surrounded by normal cells. So this is a kind of mimicking situation for the real initial stage of calcinogenesis. Then we examine what happened really at the boundary between normal and the newly emerging RAS-V12 transform cells by time-lapse movie. So in this time-lapse movie, I hope it worked. Yeah, it worked. And these cells, these two is RAS-V12 cells, and others are normal cells. So they now divided 1, 2, 3, 4. Only these 4 are RAS-V12 cells. Still looks okay, but gradually they change their morphology and moko moko, moko 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 moko, pong. Yeah, as you see, this sorry, this, maybe once more. Sorry. Okay, once more. Okay, this guy and this guy. This become a bit bad, yeah? And this bad guy looks okay, but now divide 1, 2, 3, 4. Then I don't repeat moko moko upon, but anyway, they will popped out from the society of normal epithelium. Yeah? And also, this direction is into the apical direction. So we call this phenomena apical extrusion. And also, dilation of the extrusion is quite important. For transform cells to metastasize into distant organs, they need to go basal and invade the matrix and blood vessels. In contrast, in this apical lumen, for example, in the kidney, urine is passing through, and also in the intestine, stool is passing through. So this apical extrusion is the opposite direction that is required for metastasis, and this is towards the outside of the body. So we can consider that this can be a cancer preventive mechanism. Sorry. No more. Oh, okay. And quite importantly, when CMPT stained rust cells are surrounded by rust cells, I mean, when all are rust cells, apical extrusion did not happen. So this is the most important point in a cell composition study, because this means that just activation of rust itself is not sufficient to cause apical extrusion, but being surrounded by normal cells. So the presence of surrounding normal cells is required for apical extrusion to occur. So this is a non-cell autonomous process that occurs specifically between normal and transform cells. So if you culture normal cells and rust cell transform cells separately, you will never observe this phenomenon. So this is a kind of interesting point in this cell composition study. Then next question is that whether comparable phenomena can also occur in vivo. Oh, sorry. And to examine that, we established a cell composition mouse model system by crossing these two mice. So first mice, it was Billin clearity or CK19 clearity. So Billin is the intestine-specific promoter. CK19 is the epithelial-specific promoter. And fused with LOX rasbutyrate iris GFP. Then we added low dose of tamoxifen. So under this condition, because of the addition of low dose, recombination frequency can be induced at a low. So in the various type of epithelial tissues, expression of RAS and GFP was induced simultaneously in a mosaic manner within the epithelial tissues. Then we can examine the interaction between normal and the newly emerging transforms in vivo and also in organoids as well. And just let me show you one example. So in this experiment, just we add the tamoxifen, load of tamoxifen, and after 4 days, mice were sacrificed, and intestine was observed. So this is the intestine, and so this is the control mice, just expressing only GFP. So in this mice, expressing GFP and rasbutyrate simultaneously. So in a control mice, when GFP alone was expression is induced, then just stayed in a monolayer of epithelia, as expected. But when rasbutyrate is expressed together with GFP, as you see, quite active apical aviation was observed. And this is the quantification data. So in the control condition, after 4 days after tamoxifen shot, more than 90% of cells were not extruded, remained within the epithelial layer. But in the rasbutyrate existing cells, more than 75% of cells were apically extruding or completely extruded. So this data implied that apical extrusion of rust cells indeed occur in vivo. Okay, cell composition was originally identified in Drosophila, and Eduardo Moreno's group is one of the very pioneering group that really developed this cell composition field. But recent study by us and others have shown that cell composition indeed occur in mammals as well. For example, in addition to rust, when SARC or RB2 transform cells are surrounded by normal cells, these transform cells are apically extruded. And also Scribble, Marjan, and PIF3, these are tumor suppressor protein. And when these tumor suppressor protein knockdown cells or mutated cells surrounded by normal cells, they undergo apoptosis or necroptosis. An important point is that when transform cells alone are present, neither exclusion or cell death occurs, indicating that the presence of surrounding normal cells profoundly influences the fate and behavior of transform cells. So, and also normal cells are able to actively eliminate those various types of transform cells. Okay, so this is the first part of my talk. And in the second part of my talk, I would like to answer this question. So one of the remaining questions in the cell composition field is, what is the fate of non-eliminated transform cells within the epithelial layer? Because cell composition is a kind of stochastic phenomenon, so it's not really black and white, but at certain ratio, cell composition occurs. So in a sense, under certain condition, non-eliminated transform cells could be present. So now I want to show you our unpublished data that non-eliminated transform cells are basally excluded, leading to the formation of this dome-like precancerous region. So in this experiment, using this cell composition mouse model, we examine the phenotypes of rasboutyl cells in lung epithelium. So this is a control situation. Only YFP is expressed. Then most of cells were not excluded, remained within the epithelium like this. But in this rasboutyl expressing mice, at 3 days or 1 week, most of the cells shows apically excluding or excluded phenotype. So apically exclusion phenotype was dominantly observed like this. But as time goes by, the ratio of such phenotypes decreased. And also, we never observed overproliferation of the rasboutyl cells that are eliminated apically. And actually, they seem to disappear as time goes by. So, rasboutyl cells, apically eliminated rasboutyl cells, eventually dead or eliminated from the tissue. But we realized that after 2 weeks or 1 month, such as rasboutyl cells in the basally excluded cells, RAS cells, or RAS cells in a dome-like structure were really increased. So after 2 weeks, remaining rasboutyl cells often formed dome-like structures in the lung. So let me explain you about the dome-like structure now. So this is a typical image of dome-like structure. So dome-like structure have 3 features. First is that this basally excluded RAS cells. And the second one is a bumped overlying epithelia, and also these show the multi-layered structure, multi-layered bumped epithelia, and also accumulation of stromal cells like this. So these 3 are the features of dome-like structure, and actually we define when structure has these 3 features, then we define as dome-like structure. Then first we examine what kind of stromal cells are really recruited in this dome-like structure, and we found that this alpha SMA positive fibroblasts, this is the activated fibroblasts, were really accumulated within the dome-like structure. So this shows the quantification data. Also, in addition, we found that CD45 positive immune cells are also accumulated in the dome-like structure like this, but not around the not-excluded cells so much. And in particular, this F480 positive macrophages are really accumulated in the dome-like structure. In addition, we found that in basally excluded rasboutyl cells within dome-like structure over here, so this is a magnified image, this green often lost the E-cadherin expression, whereas bimentin expression was increased. So, this and also these kind of EMT-like features were not observed in a non-excluded RAS cell. So rasboutyl expression alone does not induce EMT. So this means that basal progeon, process of basal progeon induces the EMT-like features. Okay, so this is a short summary. In lung epithelia, when rasboutyl mutation occurs, most of the rasboutyl cells were apically eliminated and disappeared from the tissues. But some of the remaining rasboutyl cells are sometimes basally excluded, leading to dome-like structure formation and the acquisition of this EMT phenotype. So next question is that whether comparable phenomena also can be observed in human. But problem is that it is difficult to distinguish between resident fibroblasts and transform cells that have undergone EMT because they look kind of similar in donor structure. So especially to examine in human pathological samples, we really need to have marker for the dome-like structure to really identify such a dome-like structure properly. Then we try to identify marker for dome-like structure. And in a previous study here, using mammalian cell culture system, we have demonstrated that expression of CD44 is upregulated in RAS-V12 expressing cells like this. And CD44 is a single-pass transmembrane glycoprotein. And also CD44 gene produces several alternative splicing variants. And these splicing variants are often expressed in advanced cancer and plays a positive role in malignant progression. However, it remains unclear whether CD44 and its variants are expressed in precancerous region. So we examine the expression of CD44 and its variants in the lung by using immunocytochemistry, by using this mouse model system. And we found that this CD44V6 antibody, so RAS-V12 cells within the lung epithelium, were specifically detected by CD44V6 antibody. And also, quite importantly, stromal cells were not stained by CD44V6 antibody at all, actually. And actually, by using the other type of specific so antibodies against other type of splicing variant, actually, we sometimes see the stromal cells were stained actually by such an antibody. So CD44V6 is a kind of specific marker for RAS-V12 expressing cells, at least in lung epithelium. And also, we found that in the mouse model system, so this basally delaminated RAS cells in a dome-like structure were stained with CD40V6 like this. So this is protection data. So CD40V6 marks RAS-V12 cells within the dome-like structure. So now we have kind of putative marker for dome-like structure. Then we now examine the human specimen. So by immunochemistry with anti-CD44V6 antibody, we examine kind of normal-looking lung and pancreas specimen. Yeah, it's very tough job actually. But after kind of extensive investigation, we found that dome-like structure-like lesions are really present in human. So this is the data of the human lung. So as you see, these are CD40V6 positive cells really are present under the bumped epithelia. And also sometimes they show the multilayer structure. And also we found that expression E cadherin was negative, although I don't show you, but also BMHC expression was positive. But the expression of cytokeratin, another epithelial marker, was positive. So indicating that these CD44 positive cells are derived from epithelial cells and have undergone partial EMT. And also we performed microdissection and genome sequencing and found that CD44V6, these cells indeed harbor the KRAS G12D mutation. And also checking pancreas, also we found the kind of comparable dome-like structures. So CD44V6 positive cells are really present under the bumped epithelium with a multilayer structure, and which was also bimentin positive and E cadherin negative. And also macrophages are quite massively accumulated. Thus, these data indicate that in human and lung, such a kind of dome-like structure-like lesions seem to be present. So we are now accumulating more samples and getting more kind of precancer dome-like structures. Okay, so this is a summary. So at least we found that in human and also mouse, such a dome-like structure-like lesion, such a potential precancerous lesions are present. And also CD44V6 can be a potential marker to identify dome-like structure, especially in human pathological samples. But still, actually, this is unpublished data, so it's still primary. So we still do not know the fate of these CD44V6 cells, actually, whether they eventually develop into malignant tumor or not. That now we are checking in vivo in my system. Okay, so this is the end of my talk. So this is my precious colleague, and also this Miho. She's an assistant professor in my lab, and Taka, actually he's a PhD student. Actually, these two are mainly working on this dome-like structure study. And these are my precious collaborators. Okay, thank you very much for your concern.